Severe inflammation in early childhood is a known risk factor for developing neurodevelopmental disorders. Now, for the first time, University of Maryland scientists have discovered that inflammation alters the development of vulnerable brain cells.
Several distinct kinds of neurons in the cerebellum mature and change over the course of childhood in ways that we had not previously known. And we found that those developmental processes in neurons are sensitive to inflammation that occurs early in childhood. The changes that we found suggest that inflammation could cause incomplete development of those neurons, and that the genes that are involved are related to neurodevelopmental disorders such as autism as well as schizophrenia.
Scientists from the Institute for Genome Sciences, the Department of Pharmacology, and the University of Maryland Medicine Institute of Neuroscience Discovery conducted the research. Using specimens from the University of Maryland Brain and Tissue Bank, the researchers examined donated post-mortem brain tissues of 17 children who died before the age of 5.
Our study relies on a new technology called single-cell genomics that makes it possible, starting from frozen brain tissue, to identify each of the individual cell types in a brain sample, understand the genes that are turned on in each of those cells, and map their dynamics.
The study is part of a landmark collection of studies describing the development and diversity of cell types in the human brain, coordinated and funded by the NIH.
The papers that are being published represent a draft map of all of the cell types in the human brain. They describe more than 3,000 distinct kinds of neurons across more than 100 brain regions. This is a landmark event in our understanding of brain cell types, but it also represents just a draft model for that. Over the next several years, what we expect will be possible is to produce a comprehensive understanding of brain cell types, how they develop, and their associations with disease.
Understanding the roles of specific cells and how they affect brain function will help researchers find treatments for a range of brain disorders. For the University of Maryland School of Medicine, I'm Larry Roberts.